everyone. So now we are going to proceed to see how ICA results might look for mobile brain body imaging data, and the fact basically that it is not directly comparable to what we know from standard stationary desktop settings. So I'm happy that Anna is going to talk about this, how to interpret ICA results, and then later after the break, how to extract events from MoBI data. Okay, Anna, please, stage yours.
Yes, welcome to the next session. I'm happy that you're all still joining and this really interesting day with a lot of content. So I'm Anna Wunderlich, and I'm one of the PhD students here at the BMobile, like the Berlin Mobile Brain Body Imaging Lab. So, I have prepared for you 2 sessions. They will be separated by a break. And so we're gonna have 1 hour about the ICA and the interpretation of independent components. And then the second part, we have some events that we can extract from the time series of the independent components. So, so where we are in the pipeline right now, from the 2 workshop days. So we're still in the ICA session that I share with Marius. And so that's where I just gonna proceed. And in this session, we're gonna look at things that follow up the independent component analysis. So maybe if you're not using that, but most of you actually stated in the poll that you do. So you can lean back and just learn something about what others do. And maybe you find something interesting that you also can apply in your analysis approaches during the event session. And so these 2 steps are not part of the pipeline itself. So the code that will be shared or that I uploaded and you may have downloaded already is separated and is a separate file, and this is just customized to the data set, but you can also just use the code lines for your analyses, and you can find the files that you're gonna need down here, and so you can already search for them alongside when I tell you what's gonna be the part of the first session. So I would be happy if you can state in 1 hour, I know the properties of the independent components, and I can separate non-brain components from brain components. So, and kind of have a feeling of what, what's basically a likely source of the activity that leads to this independent component. And for a quick overview of our audience, I would love you to respond to another poll. You already practiced that a lot. So Marius will put in a poll, and this is basically a Likert scale response to the question, how confident you are to understand independent components. So just from 1 to 7, 1 having, I have no clue, it's just nice colorful pictures, and to 7, I can understand up to 90% of all the MoBI data independent components that I have seen so far. So just for me as an information, how detailed, or I'm gonna talk in the next slides. So and in the second part, there will be some do-it-yourself part. And if you not get there, this is gonna be your homework. But I think we can manage this in time. And I'm happy that you're honest, and I'm happy that this session makes sense, because some of you just stated a 1 or a 2 or a 3. So this definitely makes sense to talk about independent components and their properties. So I'm happy to know that. And for the others that having more like the 5 to 7 responses, I'm happy to learn from you as well, because I'm just sharing what I have learned so far. And so if there are things to discuss, yeah, just join the discussion or go ahead, use the different channels to interact. Okay, so this is basically the first hour that we're going to share now. Good. Yeah, so we started with this code snippet line. This is line 32, sorry. I just added this after the feedback this morning. So you can find this in this script file and here. And so this is just a simple function that just gathers a lot of properties on one figure. So we do have, we just see like all those different properties of one independent component. So we will go or walk through all of them now in the next slides. So we always stay to this like overview, but check out like each of the properties in particular. So we start up here and oh, sorry, no, I'm reading from left to right. So so we start with the scalp map. So this is basically what you have seen like yesterday in the talks when people were talking about different distributions of activity across the scalp. And we already talked about that also this morning or lunchtime in the pre-processing pipeline, that we do see like some activity that is projected to a special number of electrodes on the scalp. And when we use the independent component analysis, we can basically go back or estimate the source localization via using a spatial filter of different weights for the electrodes, and then plot this distribution again as this nicely drawn figure. And so other examples are like this. This is like one of the participants' data that we shared. And so you can see different scalp maps. They have like some small, some like, yeah, some mixtures, some like pretty clear lines, or yeah. So they do have some really different ways to look. So some of them we're gonna just see more detailed in the later slides. So just for you to know how different they may be, and hopefully you can make more sense out of that after this talk. So the other figure that you already have seen in Mario's talk is this figure from Shirk et al. From 2019, where they just have the different dipole dipole orientation. So this is due to like different orientation of the layers of the gray matter, and how like the whole subsets of neurons are active, and how this is like folded in the brain, like in the on the top layer of the brain. This kind of defines the orientation of the dipole. So if this is like more tilted, or this is like tangential to the scalp, or if it's radial to the scalp, this like basically defines how the scalp map will look like. And so this is the scalp map going further to the ICA activation. So we do have like a scrolling part of this figure that we get if we put in this view prop function, and you can basically scroll through that, not on the slides, but usually you can, and this is something that can be really interesting and something you should put attention on is like the scale here, because you're gonna see like, for example, eye movement do have a really large scale, whereas like brain activity does have a more lower scale. And so this can be really informative about how the magnitude of this activity looks like. Below that, we do have the frequency spectra. And here you can learn a lot about power in different frequency bands, but it's not separated for bands, it's just a continuum. So, and you can, like using the function, you can define which frequency bands you want to see. And if you have like a brain activity source reflected in your independent component, you usually have like alpha peak around like 10 to 12 or 8 to 12, somewhere there. And this one is like really pronounced, but sometimes it's like not that pronounced. And yeah, and then like how this evolves above this 1/f background activity defines like different kinds of sources, and we're going to see that later on. And yeah, then we have the dipole location that was already mentioned by Marius. So this helps us to understand further like where this dipole that was plotted in the scope map is located in the brain or in the head, or if it's actually outside of the head model. This is also very informative. So for example, muscles can be located outside of the head. So yeah, so that's just the 3 different perspectives on the head, like from above, from the back, and from the side. And yeah, so in the next slide, we're gonna see a little more detailed, something you should take care of after you run the dipfit model and estimated the dipole locations. This can look like this. It's just a different way to plot that. That's also part of the GLab toolbox of dipfit. If you plot components there, you can have like all at once, or you just walk through single ones. And then you also have the coordinates that are estimated for them and the residual variance. This is what Marius already explained. This is the remaining variance that is not explained by the dipole. And so the lower this is, the more dipolar is the activity that the independent component analysis separated from the other activity in the G. So, and you should take care for that, that those are interlateral coordinates, and but actually in the EEG struct, they're the MNI coordinates. So if you use, for example, the Teleirach client, here is the link where you can find that, to get like a approximate area in the gray matter where this activity is based. Then you should really take care that in the GUI you do have Teleirach, but in the EEG struct you don't. So you may have to compute the other coordinates, or just be aware of that. Okay. So then we do have on the left bottom corner the continuous data, and this is maybe not that easy to grasp. This is a plot of the whole data set, so it's the time series of the data set, but it is plotted in like 3 dimensions. So you do have epoch data from the beginning to the end, and this is epoch now in like 8 seconds, and like every 8-second epoch is 1 layer, and then you have like all of the epochs stacked upon each other, and the magnitude of the activity you have plotted in colors. So, so what this plot basically can tell you is if activity changes over time. For example, for this component, you can kind of see a cut here, and this is due to the 2 sessions. So we do have the MoBI session and the standing session concatenated in the data. So there's actually something happening, which is surprising because it's a brainy component. And so, but yeah, basically something like that you can see, or like there are different patterns for different sources, like eye movement, or also like some muscle movement or noise just happens like at one moment in time and drives then the independent component, because the ICA is an approach that uses the whole data set and is not sensitive to just like activations that are just like in 10 minutes of 60 minutes or something like that. So there's one that remains. So as the last thing to mention in this plot is the IC label that we already started discussing, and I think it should be discussed very well and taken cautiously. That's why I also added at the end as the last property, because yeah, it is trained on stationary data, so we do see really different classifications in some cases, and sometimes if there's a mixture of different sources, it just gets confused. It's just it's hard. For example, if there is noise in the data set that is not line noise, like not 50 hertz or 60 hertz, it is not like trained for noise like other strong peaks in the data. You're going to see example later. And Marius already talked about this. So we do have those 7 classes. And this is the paper that you should read to understand how things are calculated and how this works. And something I also really like to point out is they do have a tutorial on how to understand independent components that is beyond what I'm going to share you today. So if you're interested in learning, for example, about heart or other channel noise or like some further detail, like they do have this tutorial and I can recommend it. And they do explain things in further detail. So if you want to learn more about independent components and how to interpret different properties, you should check out this. But there were also like good content shared already in the Discord channel about like other tutorials. Also a YouTube video was shared. So yeah, there's plenty of stuff where you can learn about independent components and how to interpret them. Good. So now we had this walk through the whole, I would say, 6 properties. And now I'm gonna start in the next step that we're gonna look at more the non-brain independent components first, and then also add 2 basically independent components representing brain activity. So what we're going to look at is vertical eye movement, horizontal eye movement, muscle activity, especially a neck muscle, and gait-related activity, and some noisy general things. And I put in this like figure of having a lot of noises because it's like understanding first what like other sources may be that speak up louder than the brain, if you say it metaphorically. It's good to understand them first to see like what's left and how to understand the brain as well. So it's not not all about the non-brain sources, but they're as important as the brain sources to understand our EEG data that we record in our mobile setups. Okay, let's start with the eye movement. So we do have the eye, which does have a lot of cells and neurons that start off in the retina. So there is like a strong negative pole here, and then we do have the pupil that basically has no cell bodies, and thus does have like a represents a positive pole. So if we move the eye, it's like a dipole moving in the magnetic field, and such we have magnetic field changes that we can record with our electrodes. And if the eyelid closes, the positive pole vanishes, because now like there is like current, and this way we also have a strong potential when the lid closes, and we can easily see blinks. And how this looks like, I put here another figure. That's sensor space for those 4 electrodes. And so we do see like if we look to the right and look to the left, we do have this rectangular changes in the recording. And if we do have blinks, we do see this. Just my mouse is weird. Okay, so we do have those peaks, this positive peaks in the voltage. And yeah, so as we do have electrodes located on the forehead, as well as temporal sides, right and left, we kind of collect with our EEG electrodes and EOG as well. And how this looks like in an independent component is like that. So you could basically see the same pattern that we just have seen in the EOG, in the activity, in the activations quo. We do have a scalp map that has a very frontal activity that is very high in scale. So usually it's also one of the first independent components, because the independent component analysis sorts all ICs according to how much of the data they explain. So usually those huge changes in the voltage by the eye blinks explain a lot of data in the EEG data. So that's why they turn out to be like in the first places usually. And so for the continuous data plot pattern, usually we also have this like high scale, and that's something. And also you can usually see like clear colors, I would tell it. So you see like red and blue, you can still very well distinguish. And the dipole is located frontal, and usually it would be more towards the eyes, but as the model that is underlying just uses like brain locations, it is like the brain location closest to the eyes. So this is a dependency from the model that we use for the dip fit. And what is very unique for eye components is that they have this huge power in the low frequency band. So this is something that you're gonna find in your data set, I'm pretty sure. So just for the vertical eye movement. For the horizontal eye movement, they're usually also pretty like in the front, like for the data sets we shared, they're not like the secondary one, but often it's even like IC2. So there also like a lot of activity that's due to horizontal eye movement and mobile setups. And what is known for them is that they have like also like deep frontal dipoles that are oriented horizontal. So this like the difference. So we have the right-left eye movement, and we see in the activation we do see like those rectangular patterns, and we do again have like a high scale here. And for the continuous data plot, you usually find some spot on thingies like that if there were just some moments where they did like a lot of eye movement, so that actually drive the scale here. So that's common depending on the task, like how much eye movement are fostered by the task or even prevented because of a lot of attention, like focusing on something. This will differ. And the dipole location is like again frontal, but usually independent component analysis chooses like one of the eye, or the dipole is closer to one of the eyes. But it's kind of random if it's the left or the right one. And the frequency spectrum again has a lot of low frequency power. You can see here it's a mixture of other activity as well, because we do see like this 10 Hz peak. So, and even like higher frequencies, there's something going on. So it's not super well decomposed. It's not solely horizontal eye movement. And this is what we see often, actually, for at least my data sets, that we do have a mixture of different sources contributing still to like ICs that look pretty clear. So there sometimes still parts of the data that's due to like more frontal brain activity. And this is, I think, one of the main points that also foster that speak up for the decision to not remove things from, like not to remove independent components, rather keep them and then look at what the areas of interest are and how activity changes there. Because if you remove this horizontal eye movement, you're gonna also get rid of the additional data. Yeah, so then I want to proceed to the muscle activity. So we do have plenty of muscles and around our head, in our face, in our neck, and having participants walking around mobile. They're just, yeah, they should move naturally. So they're gonna do like a lot of head movements as well as stabilizing the head, and maybe some mimicking or whatever. Like for the participants walking outside, maybe the sunshine is sometimes stronger than the shade, and you start like squeezing your eyes or whatever. So things like that happen, and we do just have our electrodes on the head, and we do have volume conduction. So it doesn't matter where the electrodes are located, actually, like they capture the EMG very well, even though there may be like the proper EMG location. But what I want to state by this figure is more or less, we just put electrodes next to our muscles. So there's gonna be a lot of muscle movement if we do have freely moving participants. So, and this is, yeah, something that we see in our data. And so what is very common for muscle activity is that they're like the scalp map is very narrow, like the locations of them are superficial, so they're like smaller than the brain component, for example, that you have seen before, where I explained the properties. So usually they're like, yeah, located and very narrow distribution across the scalp, and they're high frequency in the IC activation. So it's basically also looks like an EMG, and again we experience quite high scale but also low scale depending on the muscle that's contributing. So yeah, so this is something you can look at as well. And for the continuous data plot, we can also see like maybe there are parts in the task where some muscles are activated more than other parts. Here we can actually also distinguish the MoBI part, like where the participants were walking compared to the standing, that there were actually more muscle movements. And then for the dipole location, they're like rather to this at the surface, and sometimes they're in like really low in the neck. And this is a component we usually find in MoBI setups. So there is like kind of a brainy peak in the spectra as well, but there is like this plateau of higher frequency bands in the spectrum. And I think that was even clearer here in this first example, where you can just see like they're about like 15-20 hertz. It just goes up and remains a plateau for the high frequency bands. So this is prototypically for muscle activity. So getting towards the gait-related activity, I just want to reference this poster of Nadine. I just chatted yesterday with her, and I think we are equally happy to hear that others experience similar things when looking into MoBI data. And so this was like my first thing that I have experienced or found when I was looking for some weird pattern that I had in my mobile EEG data. And then I was happy. Oh, I thought like, well, that looks pretty similar to what I got. So maybe let's look deeper into it. And then I also found other references, but I just was so happy when I first found this poster that I just wanted to share it here, because it's related to gait-related activity. So it's about the gait artifact. Even though I would not call it my enemy, it became my friend. So, but this is something you're gonna see later in the event extraction part. So, for gait-related activity in our EEG that we record, there are some patterns that are unique for that, and I'm happy to discuss, like, because that's still work in progress. I'm pretty confident in stating it's gait-related activity. I'm not stating that it's not brain-related at all. Like, there's gonna be mixtures as well, and I'm really eager to understand this in more detail. But what I learned so far is that we do have some activation that's more central in contrast to more peripheral activity, like in the scalp maps. So this is something that happens to appear. And for the activity, we can see those patterns that have like a 1 hertz or 2 hertz frequency. So, and you definitely should take care about this pattern and memorize it and remember it tomorrow when Betty and Orosh are talking about movement, walking and gait and motion capture. So, and for the continuous data plot pattern, you see like accurately where the concatenation of the data sets of walking and standing took place. So this is like super nice. Sometimes I have those components that are just covering a part, like the beginning of the walking or like a later part. So there's differences. It's not always that nice, but usually you can distinguish the walking parts from the non-walking parts if you have gait-related activity in your brain, in your EEG activity, sorry. So for the dipole location, this is maybe drawn to this peripheral part. So I guess it's more or less driven by EEG electrode movement, actually. So there's like this peripheral part moving in comparison to the more high, like more on top electrodes. So, and for the, yeah, remember this pattern. And for the frequency spectrum, you can see like a peak for the low frequencies again, as well as sometimes I discovered a zigzag pattern, which I actually assume is due to different gait speed. And yeah, but for this data set, it's not that clear because maybe the participants were walking more or less in a steady velocity during this task. And this is another example. So here you see a different pattern in the IC activation, but you still can see like some peaks that have this one hertz frequency, but are different to eye blinks and are like very regularly during the walking part of the experiment. Okay, so I want to proceed to some noise that we can experience in our data sets. So this pattern I found, yeah, pretty often actually in different data sets that have more than 32 channels. So because then you do have like two strands having 32 channels that are actually like overlaying each other, and then they're like having some movement or some one of the channels, like one of the strands captures more noise than the other, and then you achieve this pattern that looks like intertwined with those like two strands capturing different electrodes. So if you encounter something like this, this is at least my interpretation. You can have another opinion, I'm happy to discuss. And so, yeah, I see activation is more or less narrowband, and there is like, if it's noise, there is like one peak frequency activity, but it's more high frequent than like the, for example, the gait-related activity. The continuous data plot shows more or less a regular pattern, especially also if you do have a task in front of a screen, you may experience that like a more frontal independent component like covers a lot of the noise. And yeah, then like it can even look more more regular than this does, at least what I have seen so far. And here you can see that as those data sets were processed using subline, the like line noise is like removed pretty well, but this like other noise component kind of remained. And so this is also something that's interesting for the sublime approach, that this like was not well covered, even though we tried. So so if you have the data sets, you can try and fiddle around a little bit and find out how to remove this noisy component that may be due to some setup component in this data set. Okay, another way noisy channels can look, I show you here. So this is also a pretty local one, but this is not brain, not muscle, because if you look at the spectra, it does not have this high frequency plateau, but rather as a channel that does have some noise that's recorded. And this is actually also driven by some, I would say, mechanical artifacts that disrupt this independent component activation at several times. So this is something that the ICA can dissociate from other activity sources as well. Yeah, and also for the noisy channels and noise that is captured or recorded by channels, it's still like often mixed with other activity that you can also see here. So this, yeah, should be taken in account. So this was the part of the non-brain ICs. So I would like to proceed to two kinds of independent components that represent brain activity, most likely. So we usually have some that are have a dipole moment that is radial to the scalp. For example, like the anterior singular cortex or the parietal cortex that usually have this orientation. Or we do have some tangential dipole moments like the motor or sensomotor motoric ICs that have more or less those diagonal orientation. This is like tangential to the skull. And for the radial dipole moment, I just choose again the example that I started with when we were discussing the properties in the beginning. So so what we see for brain independent components, that they are not that superficial like the noise components or the muscle components, and they are located in gray matter. If you look at the dipole position, the IC activation, I could not state any like clear characteristic here. It definitely looks different than muscle, so it's like low frequency, but sometimes you really can see like the alpha that appears several times if you scroll through the data set, and so that's pretty neat for some of the brainy components, but still there are some participants that do not show alpha that much. So yeah, for the continuous data blob pattern, this is like actually something how it usually looks, at least in my point of view. And so it's like a really more or less like even colored. It's not that clear, like the red or blue is not that clear. So I'm not sure why it actually in this data set becomes more clear in the standing phase. So this would be something that's interesting, but in my experience, this is like a usual pattern how it looks like. And the magnitude is definitely somewhere else, like compared to the muscle or to the eye movement. So we do have different scale here. Yes, so frequency spectrum having the alpha peak, this is definitely really a property that's special to independent components that represent brain activity for humans. Yeah. And this is another example. So this is more a tangential dipole moment of this independent component. So the points are basically similar, just the orientation of the dipole moment is different. And here also we cannot distinguish between the two parts of the data set. This would be more what I expect, at least for this sensor motor component. We see that there is another shape of the alpha peak here. It's more broadly, this is more white, and so this is yeah, a common spectrum of a brain component. Yeah. Okay. Well, nice. So we're good in time. So you're now the task is yours, and you can just open the data set, the VP64 filtered one, and also check out the other ones if you are fast, and compare things. And then you just find components that represent motor cortex. So they're like motor cortex, as we learned in the talks yesterday. They're located like left and right central, from the central line outwards to the periphery. And you can also look for the horizontal eye movement, like a muscle, and also find some gait-related activity. And if this is super boring for you, I have an advanced task. So you can also check for some independent components that were just like totally off classified by the IC label classifier. And yeah, so I hope you enjoy this task, and alongside that, I can answer some questions. I have just seen that there are plenty of chat contributions, so I'm a little curious what happened there.